Hello, everyone, and welcome to today's biology class, Following the Strands, a journey into DNA and analysis. I am Mr. Jack, your teacher for this session. Before we get started, please click the link that I sent in the chat and enter the code DNA 101 to join our session. We'll be using that platform throughout today's lesson for activities and responses, so once you've joined, just leave it open. This lesson is designed to help you understand two key ideas in biology: how DNA stores genetic information and how that information passes from one generation to the next. Everything we'll do today connects to those big concepts. And by the end of today's session, you should be able to describe the structure and role of DNA in heredity and explain how gel electrophoresis can be used to compare DNA samples. If you have any questions or tech issues, feel free to message me in the chat. All right, let's start with a quick warm up. I want you to answer this question. How would you describe DNA in your own words? Go to the same webpage link I sent at the beginning of class and enter up to 5 words. You have 3 minutes to submit your answers. Now, let's take a look at our word cloud together. Oh, wow, I see some great responses. The top words that stand out are genes, traits, and cell, all really important when talking about DNA. These responses indicate that you're already approaching the topic with a scientific mindset. We'll continue to explore these concepts in greater depth as the lesson progresses. Now, let me give you an overview of today's lesson. We'll start with an introduction to DNA, what it is, and how it works. Then we'll go through the steps involved in DNA analysis from collecting evidence to matching samples. After that, we'll look at gel electrophoresis and how it helps us separate and study DNA. You'll also do a short simulation to practice how gel electrophoresis works in the laboratory. Finally, we'll apply everything in a forensic science activity where you analyze DNA to help solve a case. As always, if you have any questions or thoughts, feel free to drop them in the chat. So, what is DNA? DNA stands for deoxyribonucleic acid, our body's instruction manual. It is made of units called nucleotides and shaped like a double helix, like a twisted ladder. Now, a quick question for the chat. Do you remember the base pairs in DNA? Thank you all for answering the question. Exactly, A pairs with T and C pairs with G. This base pairing is how our genetic code is stored, and it's unique to each person. Of course, except for identical twins. And guess what? That uniqueness is exactly what makes DNA so powerful in forensic science. So, how is DNA actually used in solving crimes? Well, DNA can be used to identify the suspects, link them to the crime scene, or even prove someone's innocence. Let me ask you this in the chat. Why do you think DNA is more reliable than eyewitnesses? Great thinking, class. Unlike human memory, DNA doesn't forget. Now, let's look at how DNA is analyzed step by step from evidence collection to matching samples. DNA can be collected from blood, saliva, skin cells, even hair. Now, can anyone name a place or object where you think DNA could be collected at a crime scene? Type your answers in the chat. Nice answers. Yes, those are exactly the kinds of places where DNA might be found. DNA can stick to everyday surfaces. Once collected, DNA is extracted using chemicals or automated instrument. After that, we use a process called PCR or polymerase chain reaction, which allows us to make millions of copies of the DNA. Why? Because even a tiny sample of DNA needs to be multiplied for analysis. Next, we create a DNA profile by separating DNA pieces through a technique called gel electrophoresis, which we will explore in more detail shortly. Finally, the suspect's DNA is compared to the crime scene sample. If the band patterns are the same, it indicates a possible match between the DNA samples. So, what is gel electrophoresis? It is a technique where we place DNA samples into a gel and run an electric current through it. DNA fragments move where the smaller ones go faster and farther, while larger ones move slower. The result? A series of bends that form a unique DNA fingerprint. We use this to compare crime scene DNA with suspect DNA. Now, let's have a quick check. Why do smaller DNA fragments move farther in the gel? That's right. Great job, everyone. Now, let's apply what we have learned in a real world case. A man reported a break in with valuable items stolen. Blood found near a broken window was collected, and the police identified three suspects and took their blood samples. The suspects are the delivery driver, a former roommate, and a maintenance worker. As a forensic scientist, your job is to analyze the DNA using gel electrophoresis and determine which suspect's DNA matches the sample found at the crime scene. Now, we're moving into the simulation activity, but this time you will be working in groups. I've already sent both the simulation link and the mirror board link in the chat, so please make sure you have both open and ready. You will be placed in breakout rooms for 10 minutes to complete the simulation together as a group. But before you go, I'll share my screen to walk you through the first steps so you know exactly what to do. Once you click the simulation link, you'll land on the homepage. Click Start Simulation. Then a small pop-up will appear asking you to choose a level. Please select level 1, then click Start Level 1. From here, just follow the on-screen instructions. You'll go through steps like preparing the gel, adding DNA samples, and running the electric current just like in an actual lab setup. Just a quick reminder, this simulation is for practice only. It will not show you the bands for the forensic case. Once your group is done with the simulation, move to the Miro board. That's where you'll see the real gel image for your forensic activity. You'll have 5 more minutes to work as a group and answer the questions based on that image. Let me know in the chat if anything's unclear before we start. Since there are no questions, I'll now go ahead and open the breakout rooms, and you will be automatically placed in your assigned group. Once you're in, start the simulation together, and don't forget to move to the mirror board after that to complete the case questions. Welcome back, everyone. Now let's hear from each group. Please share your screen to present your mirror board and explain your observations and your conclusion about which suspect's DNA matches the crime scene. Group 1, you're up first. Please summarize your findings based on the gel image in your mirror board. Great. Thanks, Group 1. Group 2, it's your turn. Let's hear your analysis. Fantastic observations from both groups. I noticed you collaborated well and considered the position of the bands carefully. Do both groups agree on the same suspect? All right, let's now take a look at the actual gel image. As you can see, lane 1 is the standard marker. Lane 2 is the crime scene DNA. And lanes 3, 4, and 5 are the suspects. And yes, lane 2 and lane 4 are identical, which means the DNA from the crime scene matches suspect number 2. Any surprises? Any disagreements with the result? Feel free to comment in the chat. Awesome. Thank you for your responses. Let's wrap up with a quick check for understanding using the same webpage link we used earlier. Here is the first question. What allows us to say two DNA samples match? You have 3 minutes to submit your answer. Wow, these are all correct. I love how some of you explained that matching band patterns in gel electrophoresis indicate a match, while others simply mentioned identical DNA fragment positions. Great job, everybody. Now let's move to question number 2. True or false? All people have the exact same DNA profile. You've got 1 minute for this question. All right, let's see the results. And yes, you all got it right. The answer is false. Every person has a unique DNA profile, except for identical twins. Fantastic work today, everyone. You've really shown how well you understand DNA and its application in forensic science. Any last minute questions before we end our session today? All right, if there are no further questions or concerns, that concludes our session for today. Thanks again for your energy and participation. Have a great rest of your day, and I'll see you next class.